Yo primero agradezco porque originalmente yo iba a invitar a Christophe a Morelia. Yo trabajo en la UNAM Morelia, pero el gobierno francés creo que tiene algunas restricciones para que vayan a Michoacán. Entonces pedí aquí ayuda a Adriana Garay y a Maripaz, que me ayudaron con el logística. Pues nada, gracias por el espacio para que pueda presentar. Y entonces yo invité a Christophe, que le proponía que presentara el trabajo que hace, que yo no conozco, pero este es un trabajo que hicimos juntos, y Christophe es investigador en el RDP, que es uno de los laboratorios franceses más grandes de reproducción y desarrollo de plantas. Y pues nada, pues yo trabajo con él en mi postdoc, y pues para mí es un placer que estoy aquí. Listo.
Thank you very much, Eugenio. Yeah, I would like to thank you and the university to give me the opportunity to give this presentation about cauliflowers. And as Eugenio, I think, meant at the moment in Spanish, it's not my language, you know. We worked together on this project, and Eugenio was really one of the main engines of the work. So I'm going to tell you about this. So let's start with, I hope. Okay, it's not working. Just 1 second. Okay, we'll see if this works. In case, I will do it by hand. Okay, so nature sometimes produces these remarkable shapes that have very specific mathematical properties. So, for instance, if you take this daisy, if you count the number of spirals that are made by the different flowers at the heart of this daisy, and you go the spirals clockwise and the other counterclockwise, you will find two numbers. And these two numbers, they have the property of being consecutive numbers in this series that is called the Fibonacci series, where you find one number here being the sum of the two preceding ones. Okay, and this is the property that is true over the sequence. Okay, so why plants, such as this daisy, have this property? So it's one of the very bizarre mathematical properties that a plant may have. And among all the mathematical properties, maybe one plant that has a very, very conspicuous pattern and resemble some sort of mathematical object is this broccoli romanesco. And this broccoli is actually reminiscent of a whole family of geometrical constructions that are called fractals. And the fractals, they can come from mathematical constructions, but they also can be found in nature, like this coastline of certain countries, or in vegetal and animal worlds, like this bird or this mouse. And the common point to all these objects is that similar structure can be found all over the place. Like, for instance, the cone here, the cone structure can be found everywhere in the structure, and also at every scale over and over again. And this creates this notion of complexity in the geometry object. So one of the questions that we started to investigate more than 10 years ago with François Parcy, with a colleague, a specialist in flower development, is how nature could produce this type of object that has some sort of mathematical property. And then rapidly, by discussing with François, we came to the conclusion that it would be a bit difficult to start with this object. So we would have better starting with a bit more simpler object, which is the cauliflower, the common cauliflower. And this for several reasons. The first is that this cauliflower is very close from a genetic point of view. It is in the same cultivar group as the Romanesco. They are both in the same cultivar group. The second reason is that although it don't appear to be so, the cauliflower is also a fractal object. And to convince you about that, you can just look at the spirals that are formed by the small curves that form the big curve of the cauliflower. And you have the spiral turning to the left. You can have spiral turning in the opposite direction, to the right. And beyond that, if you look locally to a small curve within the big curve, it will be exactly reproducing the same type of pattern. Spirals are made of spirals that are made of spirals. So it's basic signature for fractality. And the last reason, which is a very important one, is that cauliflower, they are actually cultivated plants, and the cauliflower itself, the curd, is the result of the transformation of an inflorescence. And this was the product of domestication. It's a long history of domestication, and the ancestral plant probably resembled this wild cabbage. And this wild cabbage is part of a huge family of Brassicaceae. And in this family, there is this famous plant that we all study in worldwide in labs, that is Arabidopsis thaliana. And it happens that 30 years ago, we realized that by making a couple of mutations in this plant, it is possible to obtain something that is reminiscent of curve structures. Okay, so this gives a good way to attack the problem. What are the genetic foundation of the construction of these very weird shapes? Okay, so now if we want to enter a bit more in the subject, then I remind you, maybe I don't know who's in the room, probably you all know how plants grow, but let's go very basically through the some definition about how it grows. So if you take a plant, the plants are growing by growing the cells at the tip of their axis in a region that is called the apical meristem. So the apical meristem will be this part here. And this apical meristem is producing laterally organs by cell division. And what you see here is that is the inflorescence meristem, the one that is edificating all these axes here, and it is producing these lateral organs. And these lateral organs are the young flowers. So this is young flower, this is another young flower. And the closer you are to the center, the younger you are. Or the later you were born. Okay, and the point is that what we would like to understand is how the genes are regulating the development of these flowers, either to produce the white flower, or if there is a mutation, to produce, instead of producing this flower, they will produce these small curves at the extremity of the window. Okay, now, how the genes are controlling this. So, I won't enter into every details because it's a super complex system, and actually in Ecole, there are groups that are working on this topic for maybe more than 10 years now. So we will have a very simple approach to this course. So if we represent here the meristem at the top and the organs laterally, that is at the top of this stem, what occurs is that there is a floration, which is a sort of chemical compound, or several types of chemical compounds that raise during the plant growth. It could be due to aging or other things. And at some point, this will reach a threshold, and upon this threshold, beyond this threshold, the plant will start to produce the flowers. The gene that is controlling whether you are a shoot or a flower is called TFL1. So TFL1 represents the shoot, the shoot identity. And on the lateral side, what you have is the flower, and the gene that best represents the flower is called leafy. So this will be represented in red and in green, respectively. So if you are TFL1, if you have the TFL1 identity, you are a in-person shoot. If you have the leafy identity, you are a flower. Okay? And this will be always the case during the presentation. Once the leafy is activated, so leafy is a sort of gate to enter the flower program. once the leafy is activated and strong, it will generate, it will call another gene that is AP1. There won't be too many actors there. 4, let's say, altogether. Don't worry about that. So leafy calls AP1, and AP1 is the beginning of the flower program. So once AP1 is launched, there is a high chance that the plant goes, this area goes to the flower. And then this creates, in the end, like through the leader, this creates the flower, the mature flower. The genes, TFL1, leafy, actually, why are they in these regions? Actually, because they are interacting. And how they interact, how the process goes, again, it's very simplified. But what we understand is that if you have the floral inducer, the F, is above a certain threshold, then if there is accumulation of a hormone that is called auxin at the tip, and the plant is able to accumulate this hormone at the tip, together, high F and high auxin, a spot of auxin, they will trigger the leafy gene. Okay. So this is represented by this. If F is active and auxin is active, then you will have a high raise up of the leafy gene. So why there is how the genes are occupying different territories? Well, because they have some interaction, and because CFL1, that is the shoot keeper, let's say, doesn't want that some flower goes into its place. So what TFL1 does is it represses the leafy as soon as it is. And leafy, in order to remain activated, will call supporting genes, genes that will reinforce the action of leafy. So it calls AP1, and AP1 will reinforce the action of leafy. So it's a very strong loop, positive feedback loop. And if the loop manages to rise up, then the flower will be initiated. Okay, and the flower will be initiated, meaning that the red identity here will be sustained throughout the growth of the lateral. Now, since AP1 and leafy are sort of self-triggering together, AP1 will have almost the same pattern as you can find on the leafy side. So you see that there is yellow where there is red, or almost everywhere. And it's not the end of the story, because AP1 needs to switch off the TFL1 so that this loop becomes a bit independent of the TFL1. So this loop will have a negative feedback on TFL1 to be sure that these regions here are not going to revert their identity to a shoot identity. Okay? Is that clear until now? More or less? Okay. I think this won't become more complex than that. So if you are there, it's okay. And in return, TFL1, which doesn't want to be swallowed by the identity of flowers, needs to repress also the AP1 gene. And in this way, you see that you have two separated classes, territories that may appear, one that is flower and one that is shoot. Okay, so these two territories, the center here and the lateral organs here, they are sort of exclusive between each other. Now, we come to the cauliflower. So with François, when we started the work, had an idea, using this type of reasoning, how cauliflower could be made. And it's very simple, actually. So if you take this scheme and you remove the AP1, AP1 is actually the gene, is representing the two genes that have been mutated in the cauliflower. Remember, the cauliflower could be obtained by just mutating two genes in the wild type. So by mutating these genes, what is left? Well, TFL1, which is here, still inhibits the leafy in this region. And because it is inhibiting the LIFI, and LIFI is no longer able, because AP1 is no longer present in the plant. So because it's no longer able to call for reinforcement, then the LIFI will rise up a bit and then will disappear, repressed by TFL1. And what François was thinking is that, okay, LIFI cannot build up here. So what will occur is probably, so it will just build up slightly, weakly, and then disappear. You see here in this organ, it couldn't be sustained. So what occurs is that TFL1 might then have the opportunity to come back in the lateral parameter instead of the flower. Okay, but what was the problem? Actually, we didn't realize in the beginning. We had to really make an even model for that, although it's obvious when you realize. If you take these actors, you see that none of them is actually pushing on TFL1. None of them is activating TFL1. So how come that TFL1 could come back in the system, whereas nobody is actually saying that you should come back in the lateral parameter? So actually one or several actors were missing from this picture. So what we did is that we studied a bit the literature. We were looking for potential candidates to switch on this TFL1. And what François, because it was really more biological work here, work on genetics. This was very much in connection with Eugenio. So what they come up with is a number of candidate hypotheses for genes that might be the activators of this TFL1. And what they found is that 3 of these candidates were actually really positive and direct regulators of this TFL1. And this was this positive regulator. So we call these 3 candidates SACs after their initials. So this stands for 3 candidate genes. And actually, this direct induction of the TFL1 was demonstrated experimentally by our colleagues, Paco Madueno and Martin Carter. And so this was demonstrated experimentally. And what we observed also is that these genes were known to induce LIFI and to induce AP1. So these SAC genes, they have a bit of double, they are playing a double game. On the one hand, they induce the shoot meristem, this shoot meristem. And on the other hand, they also induce the flower meristem. Okay? But AP1 is going to have a feedback, have a negative feedback on this sac, so this one. And due to this, you see AP1 here is active in the lateral organs, and the lateral organs, they don't have the SAC genes because of this repression. So in a way, AP1 is saying, okay, SACs, you are playing this double game, but I tell you that you are not a flower. So this is why we place this SAC gene in the shoot identity type of genes. So we have, in a way, 2 families of genes, the SACs and TFL1 genes that are representing the shoot identity, and the LIFI and the AP1 genes that are representing the flower identity. Now, if we reconsider this cauliflower salt experiment, you remove the AP1 genes that are mutated in the cauliflower. So what is left? It is left this triangular network. So as before, TFL1 is repressing LIFI. So TFL1 is repressing LIFI, which means that LIFI tends to rise up, but not so much because TFL1 wants to repress it. But SACs is pushing on LIFI and pushing on TFL1. SACs, and this we understood progressively, is not sufficiently powerful to maintain the raising of LIFI. But instead, SACs is sufficiently powerful to switch on the TFL1. So what we observe is that actually Leafy will not be able to switch on, and this is mainly because the reinforcement is lacking. The AP1 reinforcement is lacking. But Sax is here, and now is the reason why Leafy could reappear in the young primordial. So the idea is that since Sax is able to populate this region, because the F, the floral inducer, is pushing on Sax, so floral inducer is everywhere. So it pushes on Sax in the central region and also in the organs. And then the Sax is able to call the TFL1 both in the central region and in the lateral primordial. And this is the reason why then the main shoot will create lateral organs, which will transform into lateral shoots, which will transform in turn in lateral shoots. You see that there will be some sort of iterative transformation in the system. So to make sure that we understand things and that we quantify everything, we build up, and actually when I say we, it's Eugenio, we build up models in terms of ordinary differential equations, trying to capture the main behaviors of this type of model. And what we could observe, so the colors are, I think, yes, you have the colors here. Sax is blue, TFL1 is green, Leafy is red, and AP1 is yellow. So what you see is that in the shoot meristem, so in the main stem, it is vegetative, which means Sax and TFL1 are dominating. Yeah. This is a diagram that is in time. So time is proceeding here, and time is proceeding here. And this is the intensity of the gene activation. So in time, then, in the shoot, there is a rise up of TFL1 and Sax. Now, if you look at the primordial, it's a different story. Because the auxin here is activating the Leafy, the AP1 is reinforcing the action of the Leafy, and then you have this rise up of the floral genes instead of the TFL1 and Sax. Okay, now if you look in the AP1 cal mutant, which is the cauliflower mutant, what you see is that Leafy wants to, exactly as here, wants to rise up, but due to the repression of TFL1, cannot get it to the end. So actually cancels away, vanishes away, and goes back to zero. Meaning that in the AP1 cal mutant, the primordia, the lateral organs, they will have the identity of a shoot. Okay? So this network and analysis made 3 predictions. The first one is that in the AP1 cal mutant, the TFL1 activity should be high, the Sax activity should be high as well, and the Leafy should be transient in its residence. So some of these conclusions were already known in the literature, and the one that was not known is whether the Sax in the cauliflower would be high or not. So for this, François and Gabriel Tichinski, they made the transformation of these plants and attached to these soak proteins a marker that is a GFP marker or fluorescent marker. And then what you see is the activity of this soak protein in the different places. And what you see is that in the young organs here, and here, and here, there is no Sax as predicted by the model. On the other hand, if you look in the cauliflower, you see that all the organs at every edge, they are invaded by the activity of this Sax. So this was one of the confirmation of the type of reasoning that we studied. Okay, so to summarize this part, the mutant persists to create unsuccessful and successful flower, because in the wild type, what you have is different distinct regions that are populated by distinct gene activity, the TFL1 genes and the leafy genes. And the network is very stable, and the system can proceed in this way by producing different organs laterally and keeping the stem with a shoot identity. Whereas in the mutant, what you see is that the main stem is keeping the TFL1 activity, represses the possibility for the plant to go to the flower, and in the end, the TFL1 is reactivated by the presence of the side genes in the lateral genes, and this creates this vegetative structure that grows in a sort of chain reaction. But this goes quite rapidly, and the shoots are creating lateral shoots that are creating lateral shoots. Okay, and then this, just for you to know, this structure is actually a transient structure. It's not the final product of the plant. If you wait sufficiently long, it will, I don't know whether you see well, all the internodes and the elements that are produced in this structure, they will elongate. And they will produce a very huge branching structure with very many branching orders. Okay, so now we are at this point, but there was still, at least for the cauliflower, one question. Why would this part be so compact? Because here you see that the inflorescence is very elongated with long internodes. So why would this be so compact? So with Eugenio, François, and all the group, we were sort of puzzled by this, and we envisaged different things. We thought first that this could be regulated by yet other genes. But we had also another, let's say, competitive option that was that actually this occurs so rapidly that it's just the natural growth of the plant that would keep the structure compact, and because the structure that let's say the proliferation of lateral meristem goes so quick, the structure wouldn't have time to unfold and to create this one. And then this would be just due to the natural development of the plant. So to test whether this hypothesis was possible, what we did is we created a model for which we tried to get as close as possible as to the actual development in time, to the actual dynamics. So the model runs as follows. We modeled the growth of the different meristems. So what is called the apical growth. So the fact that one meristem will grow different organs, we create internode and we grow as time is proceeding. We model also the organ extension. The fact that when you have a small internode separating two leaves, it will grow in time at a certain pace. We model the lateral productions, when and how the lateral production will be created by the main apex. And finally, we model the apex identity. And for this, what we did is that we introduced in the model, it's a spatial model. And on each individual meristem, we introduced the ability to compute the state of the network associated with this system. And this is the type of model that was obtained. So this was largely done by Eugenio, where you see in, let's say, in sort of real time, not real time, this is, of course, it's not real time, it's simulated time. You see the development of the inflorescence, where all the extension of the organs and the identity of the organs is computed on the flower. Okay. And with this, what you can do, because you compute the fate of the meristem as a function of what is going on in terms of genetic interaction within each meristem, what you obtain is the wild type plant, but you can make mutations in the system. And then, for instance, for the TFL1 that produces a very short shoot with a bunch of flowers, a small number of flowers, you see that the simulation system is able to recapitulate this type of behavior. And for the so we could recapitulate the number of mutations thanks to this. And one of the mutations that is what was important is the leafy mutation, where the flower genes are absent, so you don't see any flower in the actual plant. And what you can remark, because this will be important for the rest, is that some buds in the wild type and in the leafy mutation, you have bud on this is the first order, the main stem, then these are the second order, and you see that there are buds here at the close to the leaves, and they are not developing. And this we didn't know, and actually we didn't consider in the beginning that this was super important for the understanding. So what we did in the model, that we said, okay, these buds, they are here, but we don't consider their development. So we just plugged in the system that they were not developing. Okay? So this was true for the wild type, and this was true for here. You see that in the leafy system, you don't have, in the leafy mutant, you don't have the lateral, you see all the buds here, they are not developing. It's exactly actually as in the wild type. Okay, so what we did, we said, okay, now we have a model. Let's try to figure out whether it's able to produce a cauliflower shape, because this was our original idea. So we tried this, and what we found is that, okay, the timing of the development, the dynamics of the development looks like it is possible to create these small curves. But one thing was a bit bizarre, is that here you see that in the actual plant, there are curves occurring at many, many different orders, while in the simulation, you see that you have curves on the main order 2 axis, but you don't have curves laterally. So something was missing again. And we started to wonder what was the reason of this, being given that, as I said to you, the cauliflower, if you look at the late phenotype, you see that the system has developed, has grown all the axes. So the curves are no longer curves, they are branches. And so this is the final result. And you see that you have order 3 and even sometimes order 4 and maybe order 5 that are developing. So it's not the same that we could obtain with the simulation. So something in our hypothesis was still missing. So we were considering different options and we're a bit stuck at this point because yet again, it could be yet another gene, yet another hormone signal, could be many different things. So we tried several options, and at some point, what we figured out is that if you compare the leafy mutant and the AP1 plant mutant, normally they should have the same, the primordial on the lateral side, they should have the same identity. Meaning that in the leafy mutant, leafy is the flower gene that has been mutated. Okay, so it's no flower. So the lateral shoots here, you see that instead of flowers, you have branches. Meaning that the state of the primordial that were built was of shoot identity, okay, in this plant. Now, what is the case in the cauliflower? In the cauliflower, again, we have said that leafy was trying to raise, but actually was repressed down by the shoot genes. So again, the primordial should have high TFL1 and a high Sachs genes. So if you look at the end here, the stationary state of this primordial, it's the same. And we compared these two and said, but oh, this one is branching. This one has many, many, many orders of, I don't know whether you see that just by the density of branch that this one has many orders than this one. And here the lateral, higher than order 2, they are blocked. And they should be the same actually. And actually we looked at this more carefully and we realized that it was not exactly the same in the dynamics of the system. Here, leafy is always flat. There is no leafy in the system but cannot raise. Whereas here, the leafy is trying to raise, so it reaches some certain level, before going down. And we sort of started to think, but maybe the rise of this leafy is causal to the possibility of the system to create the cauliflower phenotype, to release the higher order branches. So we made experiments, we went back to the literature, and we made the experiment, and it was also known in the literature before, that the AP1 cauliflower leafy, the triple mutant, had the phenotype that is not, so you see, this should be a cauliflower. But if you mute, in addition, the leafy, the phenotype that you are here is not like this, is not that the cauliflower. It's more like the leafy. And if you mute leafy in the cauliflower, then you don't allow this rise of leafy because there is no more leafy. Okay? And this was a key observation to say that actually there is the hypothesis that we took from this, we derived from this, is that the temporary exposure to leafy is able to release the lateral fiber. So this was giving yet another reason, yet another view to understand how the cauliflower is going. Because first, you need to understand why there is this sort of repetition of the shoot axis, one on the top of each other. But then you need to explain why they are released at all, because they were dormant in the wild type. So this was a possible explanation. And then Francois realized, actually reinterpreted one of the experiments that we did like 5 years before, and using a weak allele of a weak form of leafy in the plant that is expressed. So he forced the plant to express leafy everywhere. And because of this, you realize that in this plant, there are much more many axes that are being released than in the wild type plant. So this was yet another evidence for the fact that leafy has really something strong to do with the potential release of the lateral bud in the system. So we injected this type of hypothesis in the simulations, and then we could obtain both the fact that there is this repetition of the shoot meristem everywhere, but the fact that not only they occur on the main stem, but they occur also laterally. So maybe what time is it now? There is 10 more minutes. Okay. 8. Okay. This is a semi-conclusion before the last part. That is not too long. Yeah. Everybody is okay online now? Here with this. Yeah. So what we understood is what we think we understood is how the plant could be transformed into a cauliflower just by removing these 2 genes, and we have now these 2 reasons. The idea was because we wanted to use this lab plant in order to interpret what could be the case in the actual cauliflower that we eat. So could it be that the same type of reasons that for this transformation would be the reasons that were obtained through the domestication process? So Francois checked this by doing some genomics analysis, and what he observed, just to make a long story short, he observed that one gene, the CAL gene, was indeed muted in the cauliflower. But the other gene was not muted, was not inactivated, but it was mutated in its regulatory sequence. So the promoter. And because of this, the protein could be expressed, but only at weak levels. So in the beginning, when the plant starts to grow, this protein is almost not present. And with time, the plant is able to produce more and more of this protein. AP1 rises, and then the cauliflower will rise and make the flowers. And it's well known from growers that the cauliflowers, I mean, you must harvest them sufficiently early so that they don't grow up and make the flowers. So The story is, of course, not finished because probably during domestication, this sort of ideal schema was not the only one. The genes that were mutated were not the only one. There are probably many other actors, but this gives a sort of a way to enter into the problem of the cauliflower. Now, we wanted to go up to our initial question. Is it possible to learn something on the Romanesco based on this type of analysis? So the idea would be that if we try to transform the cauliflower here, the lab plant cauliflower, and try to figure out what could be the genetic factors or some factors, could be environmental factors, that would be responsible for the transformation, then maybe we would understand how the edible cauliflower is transformed into the Romanesco, was transformed during domestication into Romanesco. And for this, again, I will make a long story short. We came back to a paper that was produced some time ago by the group of Kiefer's group that was comparing the phenotypes at the meristem level of the cauliflower and of the Romanesco. And what they found was super interesting, this paper. It was really key for us to proceed a bit. In the cauliflower, a number of parameters, meristem size, spiral numbers, you remember the spirals, the cones are making the spirals. The plaster cone, which is the time between two consecutive organs produced by the meristem, and the organ onset delay. So let's keep on this one. They are constant in the cauliflower. They stay the same throughout the stem growth, throughout the shoot growth. Now, if they looked at it in the Romanesco, and what they found is that in the Romanesco, it's not constant at all. These parameters, they have different behaviors. So for meristem size increases, spiral number increases, plaster cone decreases. And then at this point, on the one hand, we are very excited, but on the other hand, we are very despaired. Because you see, 4 characters, you may imagine that it can be 4 different genes that are controlling these 4 different characters, especially from the fact that they were obtained by domestication. And then we started to say, okay, no, it would be impossible to conclude. But actually, what we realized, and this was like a eureka, you know, after some thoughts and discussions and trying to turn the problem upside down in all the possible ways, what we realized is actually through the phenotaxis studies, the phenotaxis models, we know that these parameters actually should be linked. And one parameter is more important than the other one, is more upstream than the other one, is the meristem size. So we said, okay, no, if this is so, maybe just by changing one of these parameters, this may, as they are all linked, maybe changing one will give the phenotype of the Romanesco. And we tried many different things. And what we finally realized is that there is one parameter that has this type of behavior. So this is a toy model that is representing the growth with constant parameters of the cauliflower. And you see that all the axes are developing basically at the same time and in the same way. So no surprise that this gives a sort of round shape. Now, if you use exactly the same model and you make the assumption that only the plaster cone will decrease, the time between 2 different organs will decrease. What you observe is that the shape will drastically change, and you will obtain this type of fractal structure that is reminiscent of the morphology of the Romanesco. So in a way, it's super simple. We just accelerated during the growth of each axis the pace at which organs were produced laterally. Okay. So, and this was the last step. We said, oh, but no, we have the interpretation. We have some sort of interpretation, some sort of hypothesis how we could go. Are we able to test this experimentally? And then I remember clearly one day, Eugenio is smiling, because we were blocked during several months on that. And one day, Eugenio came to the lab and say, okay, no, I think I know how to do it. And indeed, there is a mutation that is based on some genes actually are indeed able to increase the size of the meristem. So if you increase the size, then you will have in cascade all the other parameters grow in the right direction. So we did this. So the gene was clavata3, which is well-known genes, but for completely different reasons. And François and his group, they made this mutation in the background of the cauliflower mutant. And what we observed, so it's not as striking as would be the Romanesco shape. So in this way, we were a bit disappointed. But on the other hand, we were in a way halfway, because we obtained this conical shape, and then lateral portion of this conical shape, they are also conical, reminiscent of this notion that the fractals, they have this structure that are repeated at different scales. So, of course, it's not the final answer to the question, but obviously, and we are very confident that we are on the way towards understanding this type of things. A lot of work here. And, well, you don't know here because there is this stuff. But, there are other characters that are typical of Romanesco. You know that Romanesco, here you see it here on this side. Romanesco, if you look at them on the market, they have these fasciated stems, meristems. And you see on the plant that were grown by François and his group with the clavata3 mutation in the cauliflower mutant, we had also this type of traits that are hallmarks of the cauliflower. So it's not the end of the story, but this is what we obtain, at least. If you introduce in this system clavata3 mutation, then you obtain more conical shape. At least we are able to modify the shape of the system. And the question remains very strongly open. What are the mutations that are able to produce not only conical shapes, but conical shapes so striking ways at different scales? And this is still an open question. The clavata3 genes was not mutated, but was very weakly expressed in the cauliflower. So François made analysis, and they showed that the clavata3 was not mutated, but was weakly expressed in the mutant. So, and we are close to the end. I hope this is okay. How can we summarize this? It's about the way plants are growing. In a way, plants, they have a very simple mechanism to grow. There is a meristem, produces what I call an internode here, a piece of stem, a lateral meristem, and an apical meristem. So apical meristem produces lateral meristem, internode, and the apical meristem. If you apply this, you look here, if you apply this to this system to the growth of a plant, what you obtain is a ball, is a sort of spherical system, is a spherical ball, something that is extremely symmetric. So how do plants, they make all their architectures? Well, this type of study gives a number of hints, because what we observe is that there are two different mechanisms. One that is differentiation, the fact that a shoot meristem will give rise to a flower meristem. There may be a bit intermediate differentiation. So here we have two extremes. Okay. But this notion of differentiation is one way to modulate architecture. And then, due to this, for instance, if you, instead of making lateral shoots, you make lateral flowers, then you stop the growth of your plant. On the other hand, it's not the only mechanism. There is this other mechanism that, in a way, we rediscovered, whose important we discovered during the study, was how to get rid of the dormancy of the different buds. So dormancy may be a way for the plant to control its extent. And there are mechanisms, and actually, apical dominance, I don't know whether some of you are familiar with this type of processes. Apical dominance is a well-known mechanism. When a bud is growing, lateral buds are inhibited by the growth of the leader. So there are mechanisms in plant to control the moment where lateral buds should grow. So this is extremely important, and this was the case in the cauliflower development. And what occurs in the cauliflower? Actually, in the cauliflower, these are two locks for the growth and for the patterning of the plant. And in the cauliflower, these two locks are unlocked. They are released. The differentiation into flower is due to the inability for the plant to activate AP1. And the apical dominance, or the dormancy of the bud, it is due to the overridden the lock is overridden by the transient activity of leaf. And finally, the organ production is more rapid than internal delegation. We create this compact structure. So the plant is producing super rapidly before it elongates. This is why the system is compact. But this gives rise to fractile structure. This may explain the reason why fractile structures. And what we learned is that in the Romanesco, there may be other mechanisms, for instance, the fact that plastocone may be decreased, that would modulate the type of fractal object that we put up. So to finish, I would like to thank all the groups that have been working on this. So the main groups, François's group here, with François, Gabriel, and Marie. And this was my group with Eugenio and Etienne. And we sort of really work closely together in order to carry out all the work. Actually, this lasted over more than 10 years. And all the collaborators that were key to the different aspects of the development of this project that are listed in the authors here. And one last thing I would like to mention to you, maybe you don't know, but this work was chosen by the Academy of Science in France, and Eugenio, that did a fantastic work, and he was really fighting super discouraged during this work, received the prize of a young researcher that made what they say, grande avancée, I don't know how to translate that, in biology in the year 2021, 2022. And Eugenio received this prize from the Academy. So once again, congratulations.